My presentation was focused on immunopathology, which means that how does myeloma affect the immune system and how does immune system affects myeloma. So there's a two-way relation. And what I presented was a very significant data that shows that myeloma, which is of course an immune disease, starts because of chronic stimulation of the immune cells. So patients who have inflammatory disease, who have autoimmune disease, they are predisposed to developing myeloma. And that may be because cells are stimulated because of these indirect actions of various antigens. There's a very good publication that shows that 1/3 of the myeloma patients, their monoclonal protein is actually directed against one antigen, lysolipid. And that tells us that there are more specific stimulatory pathways. So what I discussed is two aspects. One is that there are immune cells that affect myeloma growth and survival. They promote it. And then there is second aspect, which are suppression of the normal immune responses, which would be otherwise anti-tumor, and they are suppressed so myeloma can grow. So the immune responses that supports myeloma include plasmacytoid dendritic cell, MDSCs, myeloid-derived suppressor cells, and Th17 and other inflammatory pathway-related cells. All these three cells supports myeloma growth and survival. They promote it. And that if you target those cells, you can get anti-myeloma response. So as we develop treatment, we are focusing on targeting these cells, in some case killing, say, PDCs or differentiating PDCs. On the other side, the Th17 cells produces cytokine, IL-17, IL-21, 22, 23. And one of the data that we have had from last year shows that if you use antibody against IL-17, we can control myeloma growth. Now, to give an example of IL-17, it does three things. It makes myeloma cell grow, it causes bone disease, and it suppresses the immune system. All three are done by one. And these are the three most important hallmarks of myeloma. So when we use antibody against IL-17, we can fix all those three. And so IL-17 directed study using antibody is going to start at our institution very shortly. So that's one part. The second part is that there is significant immunosuppression which prevents anti-myeloma immune responses from happening and thereby allow myeloma cells to grow. There is significant data that shows that patients who do not have good immune responses by T cells also has more growth of myeloma, progression of myeloma. So indirectly telling us that we should induce immune responses. Now, why is the immune responses suppressed in myeloma? There are many other factors, but the most prominent and better known now are the checkpoint molecules. So myeloma cells express PD-L1, and the myeloma microenvironment produces same. T cells produce PD-1, and their binding leads to inhibition of T cells. So now the efforts are to block that by using checkpoint inhibitor, PD-1 antibody, anti-PD-L1 antibody, and then you combine it with immunomodulator drugs like Lenalidomide to augment this response. And there's two clinical studies. One is pembrolizumab with lenalidomide and DAX that provides significant response, and I'll go into detail in a minute. And same with pembro-pom-dex provides similar benefit. So that's one thing. The second is we know we can do vaccination in myeloma, but with suppressed immune system, vaccines don't work well. So now we are combining vaccines, peptide-based vaccine, dendritic cell-based vaccine. We are combining them with lenalidomide and other immunomodulator to augment that effect. So all these newer methods of targeting myeloma is providing new immune responses which are very robust.